I'm a senior research engineer who is interested in bioinstrumentation and automation. I've designed several novel DNA and protein analysis instruments, and I'm passionate about molecular biotechnology. Hi, I'm Christopher Lausted, Senior Research Engineer at the Institute for Systems Biology in Seattle, Washington. I originally joined the Institute for Systems Biology because of its philosophy that in order to make big new discoveries, you needed new instruments. For example, the way the telescope opened up the heavens and redefined physics. Similarly, better microscopes, better instrumentations that let you see at the molecular level can help you understand basic biology. So the first project I worked on at the Institute was developing a DNA synthesizer based on inkjet technology, where the 4 colors of a typical printer was replaced by the 4 bases of DNA. And so we could make very rapid new designs for DNA microarrays, also known as gene chips. And gene chips for many years was the workhorse of systems biology. As a research engineer, I'm very interested in developing novel instrumentation and assays. It's a lot of fun being an engineer at a scientific institute where we don't have to mass produce products. Our ultimate product is always knowledge, research, tested hypotheses. My interest in engineering came from tinkering. I grew up on a small farm in the Midwest, and we had a lot of piles of junk, which I was allowed to take things apart and then put them back together in new ways. So my first project when I was 8, I think, was when I went out to the junk pile and I took apart some rototillers and found a Briggs & Stratton engine, and I made a little tractor. And so I would drive that tractor around and show that off to my friends, and it was a lot of fun. I always loved engineering. I always loved designing and building things, and so I studied mechanical and electrical engineering. But then in college I took molecular biology for engineers. That really changed my whole perspective on the world. Whether you're a biologist at the bench, whether you're a computational biologist, whether you're an engineer, computational skills, mathematical skills, and a little bit of coding is essential for everyone. Systems thinking is essential to all of our work, including cancer. A tumor is an entire ecosystem with many interdependent parts. We take apart these parts and then we put them back together into network models. We want to get DNA data, we want to get gene expression or RNA data, and we want to get protein data. So for this, we need high throughput DNA and high throughput RNA and high throughput protein analysis. But these machines tend to be very large. So we need to shrink it down, and then we need to combine it so that we can get all of those dimensions of the data from the smallest possible sample. I've become a systems thinker in STEM, so can you.